Hi guys, today we're going to do transcription. So we're going to explain the process of transcription, including the enzymes and the role of DNA and RNA, and essentially answer the question of how DNA is used by a cell. So we previously left off with replication, and we talked about that and discussed that, and then on Friday you should have done the transcription POGL. It's one page, so make sure you've got that done. We're going to be using some vocabulary today, the word codon, transcription. We're going to be going into the word translation, and then RNA polymerase is going to be one of our enzymes that's used. So if you need to take a second and review this vocabulary, go ahead and pause the video and review these terms. So today we're going to talk about the first step of central dogma, or the first step to make a protein. This is the process of transcription. Now when I say central dogma, okay, and make sure that you're taking a look at your notes right now, because we're going to be looking at the two sides up at the top on the upper left hand side. So the central dogma of biology basically states that DNA contains the code for making proteins. RNA, just like DNA, is made of a long chain of nucleotides that help transfer that message from DNA to a protein. So this is our central dogma. These are going to be those quotes right there on the upper left-hand side of your notes. And then this is a diagram that I want you guys to remember, because this is really going to help you when we talk about the role of DNA. So if I have DNA and DNA going to DNA, I have replication. We've been through that already. But if I go from DNA to RNA, that process is going to be called transcription. Tomorrow, we're going to be taking a look at RNA to a protein, which will be translation. But today, I just want you to focus on, if I go from DNA to RNA, that process is transcription. All right. Let's take a look at the upper right-hand side of your notes now. It says differences between DNA and RNA. So let's look at that together. Looking at this picture, what do you see are some differences between DNA and RNA? Well, I see that DNA has 2 strands and RNA has 1. I even see, looking at the bases, it looks like there's different colors, right? So when I look at DNA, one of the main differences between DNA and RNA is the sugar. So remember, Na is nucleic acid. So if it's DNA, it's going to be deoxyribonucleic acid. So the sugar difference is going to be deoxyribose for DNA and ribose for RNA. The other difference, of course, is going to be that 1 strand versus 2 strands. So DNA has 2 strands, RNA has 1 strand. And then lastly, RNA contains uracil instead of thymine. It can't carry that nitrogen base. So when we look at RNA, where you should be matching an A to a T, we're now going to go A to U. But DNA would still be the same, A to T, T to A. But over here, it's going to be A to U or T to A, T being on the DNA. All right, so we're going to practice this transcription rule. If you remembered in your POGL, that's going to be the thing that's different. So if I have a piece of DNA here, right, T will still go to A, but A now goes to U, right? C and G are still the same, T to A to U, G still goes to C, and C still goes to G, okay? So what do you notice? Hopefully you're noticing that there's no T in an RNA strand. So if I give you a set of nucleotides like this, you can immediately tell me which one's DNA and which one's RNA, because RNA won't have any of those T's. It only has the U's. Okay, when we talk about RNA, there's 3 types that we talk about particularly. This is going to be mRNA, rRNA, and tRNA. mRNA is going to be one of our most important ones, messenger RNA, because it's going to be the thing that takes the message from the DNA to the rest of the cell. It kind of looks like a thin line with like little nucleotides off of it. I think it looks like half the DNA. Okay, but its job is to read the DNA, copy that message, recopy that down, and then send it to the rest of the cell. The other part, or other type of RNA, is going to be rRNA, and all we need to know for that class, or for this class, is that rRNA is what a ribosome is made out of. So here I have a ribosome. And then lastly, we have tRNA, which is transfer RNA. And since it's transfer RNA, it's transferring amino acids to the ribosomes. So tRNA, I even think like right here, look at that. It looks like a little T, right? They don't always look like that, but I think they do. And they've got like this little amino acid hat on top. And then they have what we're going to learn later on is a codon down here at the bottom. But all I want you to know for today is that transfer RNA transfers or carries amino acids to the ribosomes. All right, so how do we make mRNA? So how does transcription occur? Well, we are going to take the instructions in the DNA. That's going to code for 4 different nucleotides. There could be 64 words that are possible, and those code words help make up proteins. So DNA to mRNA is transcription. During transcription, a molecule of DNA is copied into, or copied, let's even use the word transcribed, into a single strand of RNA. So what are the steps of transcription? To begin transcription, you have to unzip the 2 strands of DNA. You have to unzip them. So that's just like what other process? DNA replication, that's right. So the second step of transcription would be that as it unzips, you have to add complementary nucleotides of RNA, much like the base pairing during DNA replication, but now A goes to U, not A goes to T. So in this case, the strand of RNA is made of uracil instead of thymine. The enzyme that's going to be involved in this entire process is called RNA polymerase. And that makes sense because now in transcription, we're going DNA to RNA. So we have to use RNA polymerase, not DNA helicase, not DNA polymerase, but RNA polymerase. So you can see here in this GIF on the side, right? We've unzipped and here's RNA polymerase adding up those complementary nucleotides to this pink strand right here of mRNA. Here's a better look at that as well. The blue being the DNA. Here's that single strand of mRNA coming off. And those complementary base pairs, we see that the A on the DNA is going to the U on the mRNA. All right, once mRNA has read its code or rewritten it, transcribed it, the mRNA will leave the nucleus. When it leaves, the DNA will resip once that mRNA is made like nothing had happened. So if we see here in this model, the DNA, right, is unzipped. We have that single strand of mRNA being read right here. And then once transcription is done, this DNA will rezip, and the mRNA will leave the nucleus. And remember, all of this process of transcription, since we're talking about DNA, where is DNA living? In the nucleus. So the DNA is going to be here in the nucleus. It's going to go from DNA to my RNA, and then that RNA is going to leave the nucleus. And then we're going to worry about this process later, but I just want us to be focusing here today. All right, make sure that you take a look. This is a teacher's pet video. Make sure that you take a look at that in extra practice. It's really going to help you with some transcription and as we're moving into translation. So you'll see this quite a bit. If you have any questions, just let me know.